The enzyme-linked immunosorbent assay, or ELISA, is a commonly used format for serologic testing. The purpose of this animation is to explain how this test works. ELISA serologies are usually done in multi-well microtiter plates so that dilutions of serum can be easily prepared and tested. To better understand how this assay is done, let's take a closer look at what happens in one of the wells of this assay plate. To perform the assay, the wells of the plate are coated with the antigen of interest. For commercial tests, this would be done by the manufacturer of the assay. To begin testing, the wells are filled with dilutions of the patient's serum. If antibodies against the antigen are present, they will bind to the antigen fixed to the bottom of the wells. But only antigen-specific antibodies will bind to the wells. The wells are then washed out to remove all unbound antibodies. Next, a solution of an animal antibody against human antibodies is added. This second antibody is covalently conjugated to an enzyme. The wells are washed again, this time to remove any unbound enzyme-conjugated antibody. Finally, a solution of a colorogenic enzyme substrate is added. The interaction of the substrate with the enzyme on the second antibody generates visible color. The development of color in the wells with a specific antibody can be seen with the naked eye or quantified with an electronic plate reader. The reaction is less intense as the serum is diluted and the amount of antibody captured in the wells decreases. The titer is the highest dilution with definite color.
Hi guys, in this video we're going to talk about how ELISA is done. ELISA is a test that's used to detect presence of certain antigen or antibodies against those antigens in someone's blood, in some animal or in some plant. For example, this is a popular test to detect the presence of SIV antibodies or anti-SIV antibodies. That's the proper way to call it, because those antibodies are against SIV in someone's blood, and we're going to learn how to do ELISA. So let's start. So in ELISA we have wells that are coated with antibodies that we're going to check actually. So let's say we're trying to see if someone's blood contains SIV antibodies, then these wells are going to be coated with actual SIV viral particles or just some SIV virus. So these are the antigens. And then we're going to take that person's blood and centrifuge them. Now we have the red blood cells down here and the blood serum up here, and if the person has SIV antibodies in his body, then the antibodies are going to be up here, and then we're gonna put our serum into the well. So if the person is suffering from SIV and has those SIV antibodies, then those antibodies would come and bind to the well. And after this, we just wash the well, and we add secondary antibodies that are antibodies to the primary antibody. And the secondary antibody, they have an enzyme attached to it. That's why it's called enzyme linked too. So we wash the well, and after we wash the well, the antibodies they still are going to want to join to their antigen, and they're not going to be washed away. And we add these secondary antibodies, and the secondary antibodies, like I told before, they're antibodies to primary antibody, and they have enzymes attached to them. So we have this enzyme, and after this we add substrate for the enzyme, and the substrate, when being cleaved by the enzyme, it produces some color. So let's say in our case it produces some of kind of bluish kind of color, let's say, after we add the substrate. I don't know if I said this, but after we add these secondary antibodies, we wash the well again. So let's say if we don't have these primary antibodies in the first place, after the washing, the secondary antibodies are going to be gone. So this is going to produce the color, and more the color is produced, more the primary antibodies is present in the blood. So let me summarize the steps real quick again. So first we have antigen coated wells, and we add blood or other fluids from plant or animals into this, which is likely, it's not necessarily that content, but they might have primary antibody, and if they have the primary antibody, they are going to be bound to these antigens in the wall. After this, we wash the well to clean up, and after this we add secondary antibodies, which are antibodies to the primary antibody, and wash the well again. So if there's no primary antibody in the first place, this secondary antibody is going to be washed away. And after this, we're going to be adding substrate that the enzymes linked to the secondary antibodies are going to cleave, and more the color, the more the antibodies is present in the person, or higher he suffers from HIV. And this process, it's also called indirect ELISA. Indirect ELISA. And there are a couple other ways that ELISA is done, and I'm going to talk about them now. And the second way ELISA is done, it's called sandwich ELISA. And in this case, we have a well again, but in the first place we had antigens that bound to the well, but in this case we're going to have antibodies that are bound to the well. So again, let's say we're trying to see if someone has HIV, then these are going to be anti-HIV antibodies, or just HIV antibodies. When I say anti-HIV antibodies or HIV antibodies, I mean the same thing. And then we again take the person blood, centrifuge it, and put the serum in here. And in this case, it's the viral particles that are going to bound. So let's say if the person has HIV virus in the blood, then those viral particles are going to come and bind to these antibodies in our well. And after this, we wash our well. So if the person doesn't have HIV virus, then the waste, debris, other product in the well is going to be washed away. And after this, we add antibodies, another antibody to this virus that has enzyme attached to it. So let's say I add this antibody to the virus and it has enzyme attached to it. Enzyme attached to it, and we're going to wash this well away. So let's say if there's no HIV in the person, then these antibodies, they can't bind to it, and that is washed away. And after this, we add our substrate again, that's going to be cleaved by this enzyme, and the substrate, when it's cleaved, it produces some color. So let's say in this case the color is I guess it's kind of blue again. So we can see that the more the person would have this virus in his blood, the more these antibodies are going to want to bind to it, and more the color we would see. And this is called sandwich ELISA. And the reason we call it sandwich ELISA is maybe you guys could see how the antibodies and the antigen in middle, they kind of make like sandwich. And so this is sandwich ELISA. And the third type of ELISA that I'm going to be talking about is called competitive ELISA, and this is how it's done. So in this case, it's again the antigen. Let's say if we are trying to test for HIV, then the HIV viral particles that are bound to the well, and we take a person's blood and centrifuge it. So again, the red blood cell, the blood particles, red blood cell, they settle down, and we have this serum up here. And this serum, if the person is suffering from HIV, is going to have HIV viral particles. And then we put the serum in another vessel, this vessel would have a known concentration of antibodies against the HIV virus. So if the person has HIV virus, those virus are going to bind to these antibodies in here. If they don't, then they're not going to bind. Now we dump up the content of this vessel to our well. That's what we do next. And when we dump it, now you can see that these antibodies, they could bind to the viral particles like from the blood. They could also bind to viral particles in the well. So more we have viral particles in the well, these antibodies are just going to be floating here, being bound to the viral particles or the antigens. And let's say there could be some free antibodies here too, because we don't have enough virus, let's say. Then these antibodies would be bound to the well. So less the virus we have, the more these antibodies are going to be bound here. But the more the virus we have, less the antibodies are going to be bound to the well. And after this, the well is washed away so that these things get off. And after washing, we're going to add secondary antibodies to these primary antibodies, and they have enzyme attached to it again. So this is how it looks like. And after this, we're going to wash our well again so that some of the secondary antibodies that are not bound to these primary antibodies, they could be washed away. And after that, we add substrate to the enzyme attached to the secondary antibody. And let's say in this case, when the substrate is clipped, it makes some purple color. And this is called competitive ELISA. So as we can see in this, what happens is the less the person has virus in his blood, the more these antibodies would bound to antigens in the well, and more the secondary antibodies would bind to them, and more the color would be here. So less the virus the person has, the more colorful will be the well. In this case, more the virus the person has, or more the antibodies against the virus the person has, more colorful the well would look. So this is indirect ELISA, sandwich, and competitive ELISA. I hope you guys learned from my video. Thank you for watching my video. Bye.